Looking deep into the rock on the atomic scale tells us a lot about what environmental conditions were like at the time of the late Devonian mass extinction. This science is called geochemistry. We use chemical analyses of rocks that were formed in ancient seas to understand what the oceans were like during the late Devonian. All life on Earth is carbon-based. So when organisms living on the ocean die, their bodies, and thus the carbon that makes up their bodies, are deposited on the seafloor. We can then go and examine different properties of that carbon that get preserved in rocks to learn about that life and the environments in which those living things lived. Using geochemistry, we can find information about the past in rocks, even when there are no obvious fossils of things like a shell that exist. Subtle changes in ocean temperature, salt content, or even the abundance or lack of life in the oceans are captured at the atomic scale. By taking samples from all around the world and applying these modern techniques, we are able to better understand what unfolded at the end of the Devonian. While we usually think of fossils as being things like bones and shells that we can see with our eyes and hold in our hands, much of the fossil record is actually made up of microscopic fossils of single-celled organisms, things like amoeba, yeasts, and algae. Even though these organisms are tiny, many of them do get preserved in ancient rocks, and we can use chemical techniques to dissolve the rocks around them and find the fossils. In the Devonian, one of the really cool things we've discovered is that there are microscopic fossils preserved in rock that were deposited during the mass extinction when there are no animals or plants preserved. We are uncovering what these tiny fossils can tell us about the extinction.
Before we can look at rocks under the microscope, we need to cut them into incredibly thin slices so they're nearly transparent. These slivers of rock are mounted on a glass slide so that geologists can look at the textures and structures of the rock under a microscope. One of the many types of minerals that we look for are called pyrite framboids. These are tiny mineral clusters that look like raspberries. Framboids form when there's very little oxygen in the water column, so they can be useful for tracking oceanic oxygen loss. If you have a rock with lots of framboids, you know that the ecosystems that these rocks were formed in were dealing with oxygen deprivation. If there's no framboids at all in a sediment, it means the water column had plenty of oxygen and the ecosystem was healthy. Most of the minerals we look at in our rocks are too small to see, so we use a process called X-ray diffraction, which identifies minerals based on how X-rays pass through them. It's important to know what minerals are in the rock because they tell you about the environment where the rock was deposited. We can use cathodoluminescence, called CL for short, to tell us about how the minerals that make up rocks may have been altered by fluids passing through them. You hit a rock with energized air and it glows in ways that shows how the crystals have grown, or more importantly, if they've been damaged and then healed. If the fluids have damaged the rock, usually they alter the chemical signatures, and then we know we probably can't use that data when we look for patterns in the chemistry. We can use certain metals and weird-sounding elements that are not very common to tell us about oxygen loss and changes in biological activity in oceans. For example, molybdenum is a really unusual element in most rocks, but during time periods when there's significant oxygen loss in oceans, you can find molybdenum concentrated in the sediments. Most of the time, there's very little of these elements in most sediments, but during mass extinctions, they can get concentrated.
Stable isotopes of elements are where elements have an extra neutron in their nucleus, but the same amount of protons, which makes the element a little bit heavier than it would otherwise be. This difference in weight means that the elements that are a little bit heavier tend to get concentrated when certain events occur, such as a mass extinction or major changes in climate. For example, water has oxygen, H2O, and the heavy oxygens with an extra neutron don't evaporate as easily as the lighter oxygens, which don't have that extra neutron weighing them down. We can use these trends and these oxygen isotopes preserved in rocks to track climate changes through time, like when there were wetter versus drier periods, or colder periods with glaciers versus warmer periods without glaciers. Taken together, these chemical and mineral fingerprints that we analyze in the lab are essentially the equivalent to the trace evidence of crime scene investigators. But in our case, we have the difficult task of looking for a natural disaster of epic proportions that happened hundreds of millions of years ago.